Hey y'all, welcome to chapter 4.6. This is the last section of chapter 4, so this is the last lecture for chapter 4. So we talked a little bit about how things cross the membrane in chapter 4.5. In chapter 4.6, we're going to talk about how we bring lots of stuff across the membrane. So we're going to talk about a process called endocytosis, specifically phagocytosis. Okay, so our section objectives for 4.6 is we need to be able to describe how endocytosis and phagocytosis transport material into a eukaryotic cell. We need to be able to explain how pathogens can take advantage of endocytosis and phagocytosis to gain access to the cell. Okay, so I have a question for y'all. How does the cell get really large molecules across the membrane? Because in the previous lecture, we talked about things directly crossing the membrane by diffusion. We talked about channel proteins or transmembrane proteins that are going to allow things like polar and large molecules and charged ions to get across. But what if we have something really big? Okay, that's bigger than what our channel proteins will allow across. How does the cell do this? Well, the answer is a process called endocytosis. Now, if you remember, endo means in, cyte means cell. So endocytosis means into the cell. Okay, so there's a lot of different types of endocytosis, but in general, endocytosis is a form of what we call active transport. So it's going to use energy. That's going to move large molecules and large pieces of other cells and debris into the cell by enclosing them in something that we call a vesicle that we make out of the plasma membrane. Okay, so even though there's several types of endocytosis, they all have the same basic process. Okay, so the first thing that's going to happen is that the plasma membrane of the cell is going to fold inward. We call this process invagination. So the cell is going to fold inward and it's going to create this little pocket. Okay, this little pocket's going to be a place that the target particle or particles, so these molecules that we want to bring into the cell, this is where they can be surrounded by the membrane. Okay, so the membrane's going to come closer, comes closer, and we're going to truly make a pocket. And at one point where they're almost touching, some specialized proteins are going to come in and they're going to pinch off this piece of the plasma membrane with our molecules that we want to come into the cell in it, and it's going to create a little pocket called a vesicle. And that vesicle, sometimes also referred to as a vacuole, this vesicle is then going to come into the cell, and then what happens at that point depends on what type of endocytosis is happening. But this process of invagination happens in every type of endocytosis. Okay, we have three specific types of endocytosis. We have phagocytosis, pinocytosis, and receptor-mediated endocytosis. So we've talked about all these before if you've taken biology at the high school, but in this lecture, we're going to specifically talk about phagocytosis and receptor-mediated endocytosis. Okay, we're not going to really go over pinocytosis. One of the first things we need to do, we can always tell what's going on if we know the roots of the words, right? Remember, cyte means cell. Well, phago means to eat. So phagocytosis literally means cell eating. Pino means sipping. So pinocytosis is cellular sipping. And then receptor mediated means that the proteins in the membrane are going to trigger endocytosis. Okay, so pinocytosis, this is a way we can bring in little amounts of things. Okay, hence the cell sipping part. Okay, but what we really want to focus on is phagocytosis and receptor mediated endocytosis. Okay, so let's start with phagocytosis. Okay, this is the way cells bring in large particles. Okay, so this is going to be like other cells or cellular debris. So if a cell has ruptured or been broken down, is old and defunct, a lot of cells are really great scavengers. So what they're going to do is they're going to bring in this cellular debris, and they're going to bring it in by phagocytosis. Okay, so we're going to have the membrane surround the particle. It's going to create that pocket through that process of invagination. Okay, it's going to pinch off and make it into a food vacuole or a vesicle. Okay, usually they're used interchangeably. I'm going to use the term vesicle. Okay, so once this happens, this cellular debris comes in. Okay, the membrane that forms the vesicle is going to be stabilized by a protein called clathrin, so it doesn't break apart while it's traveling through the cell. So that vesicle is going to come in and it's going to fuse with a lysosome. Remember, lysosomes from biology are specific to animal cells. We don't find them in plant cells. Okay, so the lysosome we refer to as the garbage disposal of the cell. So lysosomes have this acidic interior. So they have what digestive enzymes, they have acids, that their whole job is to break things down. So our vesicle is going to come in that contains what we want to bring into the cell, and that vesicle and the lysosome are going to fuse together. What this is going to do is it's going to dump all of these digestive enzymes, this acidic environment, into the vesicle, and those enzymes and those acids are going to start to break down that cellular debris, and we're going to break it down into components that our cells can use. Okay, we're going to break down proteins into individual amino acids and recycle them. We're going to break down starches and things into individual sugars that we can then use to make energy. So this is one of the ways that our cells can get what they need. Okay, so I have some videos here. These are really important. Okay, so the first one we're going to watch is actually really important. This is how our immune system functions. Okay, so phagocytosis is our immune system's main line of defense. Okay, so we have a neutrophil here. Let me get my, okay, we have a neutrophil here, and we have a bacteria. This is actually the Staphylococcus aureus bacteria. So this is a bacteria that is going to cause pneumonia. Okay, so our immune system are going to look at those receptors on the outside of the cells, outside of the cell membrane, those proteins, and it's going to look for proteins that it recognizes. So if it recognizes something like self, like these red blood cells here, okay, we can see that the white blood cell doesn't really care about these red blood cells because it recognizes them as being part of you. But what it does realize is that this bacterial cell, the Staphylococcus aureus, is not supposed to be there. Okay, so once it recognizes it as non-self, it is going to pretty much seek and destroy. So as we watch it, it's going to chase it, and there's going to come a point where you see the white blood cell invaginate and phagocytose that bacterial cell. Okay, that bacterial cell will then be in a vesicle, and it's going to meet up with lysosomes, because white blood cells have lots of lysosomes, and it's going to break it down and it's going to get rid of that infection. Okay, phagocytosis is the main way our immune system functions. Okay, so a lot of single-celled organisms like amoebas use phagocytosis as a way to get nutrients. So in this video, we can see our amoeba, that's the bright green guy, trying to phagocytose a yeast cell. Okay, so you can see it making a pocket, trying to bring it in. Well, there's a little bit of a problem here. Our yeast cell is considerably bigger than our amoeba. Okay, so our amoeba is going to try in a different direction. So you can see the pocket forming when it latches on. Okay, so the pocket's forming. It's trying to eat it. Okay, unfortunately for this amoeba, it is not big enough to fully engulf this yeast cell. Okay, but you can see the pocket forming is trying to fold it in and bring it in as a vesicle so it can break it down into the nutrients and building blocks that it needs to function. Okay, at some point this amoeba is going to give up, but this is a really good illustration of this invagination process. Okay, so one of the last things we're going to talk about is a case history. This is the last case history for chapter 4. It is called Chicken Surprise. So our case history is going to revolve around Kelsey. So it's October 2013, and Kelsey is working as a computer programmer for a startup tech company in Silicon Valley. Okay, she's 28 years old, she's single, she's just had a long day of work. Okay, so Kelsey goes home after work, and in the middle of the night, she wakes up with the most terrible abdominal cramps. Okay, the cramps progress and into diarrhea, and shortly after, she gets a fever and a terrible headache. Okay, so she just thinks she's gotten the flu or something. So she just waits it out, and 3 days later, her fever has gone away, but her diarrhea has continued. In fact, it's getting worse. Okay, her diarrhea is so severe, she's going about 20 times a day. Okay, and this continues for a few more days. But the thing is, after a few days, her stool is starting to show blood. Now, whenever you have blood in the stool, this is a bad sign. You need to go see a doctor. Okay, so Kelsey obviously is freaked out, so she immediately heads to the ER. Okay, when she gets to the ER, she's admitted. Okay, Kelsey is extremely dehydrated, so because of the diarrhea, so she's put on IV fluids. Okay, so when the attending physician for the ER comes in, he starts asking her a bunch of questions, like, have you traveled? And one of the things that the physician asked is, what was her dietary history? What has she eaten before symptoms started? Okay, so Kelsey, being single, didn't want to cook after work that day, so what she did is she stopped at the supermarket and picked up a rotisserie chicken, and she ate the rotisserie chicken for dinner the night that her symptoms started. Okay, so armed with this information, Kelsey had a stool sample and a blood sample that were collected from her. Okay, they ran it through an antibody test with fluorescent microscopy. Well, what the doctor was thinking it was happening actually did happen. So the antibody test came back positive for Salmonella enterica cerevar Heidelberg. Okay, so just a quick note, cerevar means strain. Okay, so this is a specific type of Salmonella enterica. So Salmonella enterica is actually a pretty interesting bacteria. It has over 4,400 different strains. Okay, unfortunately for Kelsey, the strain of Salmonella that she got, this Cerevar Heidelberg, is one of the more terrible strains. Okay, so it's if you have to get Salmonella, this is not the one that you want to get. Okay, but both her stool sample and her blood sample show that she has this infection. Okay, because Kelsey's symptoms are so severe and the antibody test came back, she was immediately treated with antibiotics. And because she was so dehydrated, she was immediately started on rehydration therapy. Okay. Ultimately, Kelsey was not the only one that showed these same symptoms. Okay. In fact, Kelsey was one of more than 400 cases over 23 states that had contaminated rotisserie chicken. Okay. So they traced back these chickens, and all of these contaminated chickens came from one single California farm. Okay, so between the antibiotics and the rehydration therapy, Kelsey gradually recovered. But because this was such a terrible strain and they were worried because her symptoms were so terrible, Kelsey came back 3 months later for a checkup. But even though Kelsey had recovered, this Heidelberg strain of Salmonella was still detected in her stool sample. Okay, so my question to you is, how can Salmonella survive 3 months post-antibiotic treatment and post-illness? Kelsey's recovered. Why is there still Salmonella in her samples? Well, the answer has to do with endocytosis. Okay, so Salmonella enterica is going to trick your cell into bringing it in via phagocytosis. Okay, but Salmonella enterica is going to make some proteins and stuff that's actually going to stop the process. So it's going to make it so that the lysosome is not going to fuse with the vesicle that's come in. Sometimes we refer to the vesicle in endocytosis as the endosome. Okay, so the vesicle that has brought the salmonella in isn't going to fuse with the lysosome, which means that the salmonella enterica is not going to be broken down. Okay, what this means is that the salmonella now has a nice little home and a hiding place from our immune system. So it's a very safe place. And one of the reasons why Kelsey keeps having this bacteria show up in her stool samples even months later is because that as it slowly breaks out of the vesicle, it's going to cause an infection and it's going to still be there. Okay, it has created a way to hide itself. Okay, where it's safe from antibiotics, it's safe from your immune system, so it can grow and thrive in these vesicles. Okay, in fact, the process of salmonella hijacking your vesicles, your endosomes, is a method of pathogenesis. So remember, pathogenesis is how pathogens cause disease. So this is one of the ways that they can continue to cause disease. Okay, the last thing we are going to talk about in this chapter is really important, and this is the main way that viruses and bacteria can enter your cells. And it's called receptor-mediated endocytosis. It's very similar to phagocytosis, but we're going to use those protein receptors on the outside of our cell membranes. Okay, so receptor-mediated endocytosis is going to be a way to bring in specific types of molecules. Okay, so the way this happens is that there's going to be a cluster of receptors in a certain region of the plasma membrane. We call these clusters coated pits. Okay, this is because on the inside of the membrane where these pits are, the membrane is coated with a layer of proteins called coat proteins, like clathrin in the previous example. Okay, so what happens is what these receptors are in a cluster. So they're specific because they're proteins, they're specific for one thing. And when a lot of the receptors in this cluster have bound the substrate, so this molecule that the cell wants to come in, it's going to signal the cell to start the process of endocytosis. Okay, so they're going to make the pocket, so invagination is going to happen. We're going to make the pocket. Okay, we're going to bring in not only the piece of membrane, we're going to bring in the receptors. Okay, and what's going to happen is these coproteins, this clathrin, is going to help to keep the vesicle stable when it comes in. Okay, so it's going to give it its round shape that easily buds off from the main membrane. Okay, and it's going to allow our cells to take up large amounts of a specific molecule. Okay, so a lot of these are going to be relatively rare in the extracellular fluid, so that means they're going to be low concentrations. So sometimes it takes a minute to fill up these receptors. Okay, so it's a way to bring in useful things. But unfortunately for us, a lot of viruses and bacterial cells and toxins have evolved that they can have a similar shape as the molecules these receptors are binding. So when this happens, instead of binding what your cell needs, it's going to bind the virus. And when it's enough virus and molecules have bound to these receptors, it's going to signal to the cell that it needs to bring this coated pit in. So flu viruses, diphtheria, HIV, COVID-19, the cholera toxin, like we talked about in the first of chapter 4, these all use this receptor-mediated endocytosis to gain entry into your cells to make them their host. Okay, so this is one of the most used versions of endocytosis that pathogens use to gain entry. Okay, so that is chapter 4.6. That means we're done with chapter 4. Okay, so that's it. Chapter 4 homework is going to be due pretty soon at midnight. Check about said classroom. Check the Canvas page for the due date. But before we end this lecture, let's finish one more review question. Okay, so review question number 2. I want you to answer this. So determine which would occur when a mammalian or animal cell is placed in a hypertonic environment? A, there will be no net flow of water in or out of the cell. B, the cell will burst because large amounts of water will flow into the cell. C, the volume of the cell will shrink because water will flow out of the cell. Or D, the answer cannot be determined by the information provided. Okay, so take a minute, pause the video, and see if you can't find the answer. Okay, so this is a vocabulary question. We need to know what hypertonic means. Remember, hyper means high concentration, or high. Tonic means a solute concentration outside the cell, so this is a high solute concentration. So remember, water goes from an area of low solute concentration to an area of high. So if the concentration of the solute is high outside the cell, the water is going to leave the cell and flow out. Okay, so the answer to this question is C. Now, this is a good question to know, say for like an exam, because this is one of those questions that can easily involve 3 different vocabulary words. Okay, I could easily change it to hypotonic, and the answer is there. Okay, so this is a good type of question to know because it can be changed easily. Now, that being said, when you see questions like this, even though we've seen this specific one in the lecture, make sure if you see a question like this on an exam that you read it carefully and you're answering it correctly, because hypertonic and hypotonic aren't that far from each other, okay, which could really screw up your answer if you go to answer it just like this question instead of what it's really asking. Okay, so that is chapter 4. We'll see you for chapter 5 in the next video.